students, welcome back to my channel. Today we are going to learn all the experiments from plant physiology photosynthesis part. All the plant physiology experiments are important for you to score the diagram based question from part B of your question paper. If you're not clear with the parts of the question paper or the pattern of the question paper, please make sure that you watch that video that I've already shared with you, clearing all the important points regarding the pattern of the question paper, how the questions are going to be framed, and so on. So I'm going to add that in the description below as well. If you have not been watching all the important videos also, please do watch, please do subscribe to my channel, and let's learn experiments in photosynthesis today. So first and foremost, the question that is asked is, why do we de-starch a plant before we start a photosynthesis experiment? For that, we should know what is meant by de-starching, isn't it? So what exactly is de-starching? And when we de-starch a plant, what exactly are we doing? All right. So before starting any photosynthesis experiment, you should know that the end product of photosynthesis is starch itself. Therefore, to know whether photosynthesis has occurred or not, we can look for the amount of starch present, right? Therefore, we need to remove all starch before starting all the experiments on photosynthesis, right? That's why we de-starch a plant. How do we de-starch a plant? We need to keep the plant in darkness for 24 to 48 hours. When we say we are de-starching a plant, can we remove starch from the entire plant? No. Okay, we can remove starch only from the leaf. So when you get an MCQ asking, when de-starching is done for a plant, which part are you actually de-starching? You are de-starching the leaves. Students, in all the experiments that you learn, experiment number 1 to show chlorophyll is necessary and so on, in all the experiments they'll tell you to do something called as do the starch test. Do the starch test is given. So what is exactly is this starch test? We should know. Under this, a lot of MCQs are also to be expected. So let's understand the starch test. Okay, so what are we doing? We are checking for presence of starch. Starch is a polymer of glucose and it has a helical structure. Because of that helical structure, it can bind to iodine, giving a blue-black color. That is, if starch is present, when you add iodine to that leaf or whatever you're testing with, it should give you blue-black color. Not blue, not black, blue-black color. If starch is not there, that means it will just be brown itself, which is the color of iodine. Alright. So what are the steps to do iodine test? First, you need to take the leaf, boil it to kill the cells. Underline it, learn it, MCQ. Then, this leaf you are putting in spirit, that is methylated spirit, to remove chlorophyll. Second step. Then you need to add it into hot water. Then you are adding and cool it down and then add iodine solution which is brown. If starch is present, the part where starch is present will turn blue-black. That is called a starch test. Okay, now let's go and learn all the experiments. First experiment, we are trying to prove that chlorophyll is required for photosynthesis. Okay, so in a plant, we take one leaf and we need to select a plant which has some parts where green color is there. That is, other plants are not having any chlorophyll on it. You probably have seen such leaves. Some leaf where light yellow color is there or purple color is there. Such leaf we have to take. And in that leaf, wherever green color is there, we know that chlorophyll is there. That's why it is green. Alright. And we are checking if that is very much required for photosynthesis or not. Okay. We're checking chlorophyll is required for photosynthesis or not. For that, what do? We take the plant and first and foremost, we de-starch it. That means we keep it away from the sun in darkness. Then, so when we are de-starching, what are we de-starching? We are de-starching the leaf only. Remember this, even though we are keeping entire plant in darkness, we can de-starch only the leaf. 1 mark MCQ. Alright, now once that is done, we need to select one leaf from it and add, so obviously we need to, after de-starching, we need to keep it in sun, right? Only then photosynthesis will occur. After that, we need to select one leaf, and that leaf we are checking for presence of starch or not. If presence of starch is there, photosynthesis has occurred. Okay, so we'll take one leaf. In that leaf, wherever green part is there, when you add iodine solution, we just don't add, we do this entire test. Okay, then what should we find? Wherever green color is there, that is wherever chlorophyll is there, starch would have been produced. Therefore, it will turn blue-black. Wherever green color is not there originally on the leaf, yellow color is there, purple color is there. That means chlorophyll is absent. Photosynthesis did not happen there, right? Therefore, that part will not turn blue-black. Instead, it will be brown only. So that is the end result of it. Okay. From here, another MCQ that comes is, give an example of variegated leaf plant. Coleus, geranium, croton comes as 1 mark question. Variegated means what? It means that some part of the leaf is green and some part is not. That's it. Coming to second experiment. What are we trying to prove here? We're trying to prove that sunlight is required for photosynthesis. How to check that? We need to have a leaf. Same method. We need to have an entire plant. We need to discharge the plant. We need to bring it back to sunlight. But before keeping the entire plant in the sun, a particular leaf we are selecting, that leaves certain parts we are hiding using black paper from the sun. That means we are taking a cutout like so, black paper, and covering one leaf with it. What are we actually doing? On this leaf, we are creating two areas. One area where sunlight is falling, one area where sunlight is not falling. That means wherever sunlight fell, photosynthesis would have occurred, resulting in production of starch. If we do starch test or iodine test on it, that part where photosynthesis occurred, starch produced, where sunlight fell, that part is going to turn blue-black. Other part which was hidden from sunlight, no photosynthesis would have occurred, correct? Therefore, that part will not turn blue-black. Instead, that will be brown in color. That's the end result of it. In experiment number 3, what are we trying to show? We are trying to show that carbon dioxide is required for photosynthesis. We know that carbon dioxide and water vapor are very much required, or they are the raw materials for photosynthesis. Okay, so how to prove that? For that, we should have a situation where one part of the leaf is not getting any carbon dioxide and the other part is getting the carbon dioxide. Okay, so how to create that? First foremost step, we are de-starching the entire plant. That is, we are de-starching only the leaf, keeping it in darkness. After that, what are we doing is, we take a conical flask inside which potassium hydroxide is present. What is so special about potassium hydroxide is, it has the ability to absorb carbon dioxide. Okay, so, and then into that we are inserting one leaf half part. Half part of this leaf is inserted, tightly corked so that no other carbon dioxide from outside can enter inside this. So we have created one leaf part where carbon dioxide is not accessible to it. Okay, that is what we have done. Now the rest of the leaf is outside, which is getting carbon dioxide. Now the entire setup is kept in sunlight. So the plant is having enough sunlight, but one part of the leaf which is inserted inside the conical flask will not get any carbon dioxide, as this carbon dioxide is already absorbed by potassium hydroxide. the chemical. Alright, now we take out the leaf and we do the starch test or the iodine test. What do we have? What will be the result? The place which was inserted in the leaf which was inserted, half part way which was inserted, will not show blue-black color with iodine, correct? Because no starch would have produced in that, because no photosynthesis would have occurred, right? But the part of the leaf which was outside, which got enough carbon dioxide, which got enough sunlight, that part would have underwent photosynthesis and would have produced starch, which gives blue-black color. Okay, so that's the next experiment. The last experiment, number 4, is we are showing that oxygen is produced. We are not showing that oxygen is required, because oxygen is not required for photosynthesis. We are showing that oxygen is produced during photosynthesis. For this, 1 mark question comes, name any aquatic plant used for this experiment. 1 question possible. So answer is hydrilla or elodea. We can answer any one. Okay. So for this experiment, what should we do? We should have a setup where any aquatic plant, either elodea or hydrilla, are kept inside a conical funnel. Funnel is kept inside the beaker with some water. Normally, the plant is there in water, so it's going to photosynthesize, isn't it? Producing oxygen. But we should have that oxygen with us to check whether oxygen only is produced or something else is produced, right? So we need to collect that. So what do? First, we kept a beaker, water is kept. Inside that, we kept the hydrilla plant. Hydrilla plant is covered by this funnel, this one, this part, right? Now, oxygen will be produced normally because of photosynthesis. We need to collect it to check it. For collecting that, what do? We take one test tube and invert it upside down. We are keeping on the opening of this funnel, so that when this plant is getting sunlight and doing photosynthesis, it produces whatever gas it's producing, and collect it. Okay? So that way we get this gas collected in the test tube. To check whether it is oxygen only, we can bring a splinter near it and it will burst into flame. We know that oxygen helps in burning. We have learned that in smaller classes. So hence we can prove that oxygen is produced during photosynthesis. I hope all the experiments are clear to you. If not, please connect with me through the chat box. Thank you.